Hello, and welcome back to cellular respiration. This video we're going to talk about the Krebs cycle and what products are generated out of the Krebs cycle. I've got a handy little list over here. All right, we've got 2 carbon dioxide, 1 ATP, 3 NADH, 2 FADH that are going to be generated per turn of the citric acid cycle. Okay, one thing to keep in mind, we get 2 turns of the citric acid cycle per glucose. So if I ask you how much of each product are made for 1 glucose molecule, you would double each of these, okay? So we would end up with a total of 4 carbon dioxide, 2 ATP, 6 NADH, and 2 FADH per glucose, okay? The ones in yellowish green here, this is per turn of the citric acid cycle, okay? So what we're going to have, we talked last video about the generation of acetyl-CoA, and that this acetyl-CoA is going to be what primes our citric acid cycle, okay? So what that's going to do, that's going to enter our citric acid cycle, all right? And what we're going to have given off out of this, our CoA is going to be clipped off of that molecule. And what's going to happen, those 2 carbons that came in as part of our glucose molecule that I have in green here are going to attach to a 4 carbon molecule that is already in the citric acid cycle. That's going to be oxyl acetate. I want you to know what oxyl acetate is. It remains in the citric acid cycle. It gets reused and regenerated each turn of the citric acid cycle, okay? So our 4 carbon oxyl acetate gets joined to the 2 carbons that came in as part of our glucose molecule. And what that generates our citric acid. That's why this is referred to as the citric acid cycle, okay? So that's going to be our first step, all right? Then what's going to happen, what we're going to do is we are going to remove each of these carbons to generate some other products and generate our NADH, our ATP, and our CO2, okay? So if we lop off, because now we've got 6 carbons, all right? What's going to happen, we're going to lop off one of those carbons. That's going to generate alpha ketoglutarate. That's going to be a 5 carbon molecule, all right? Because we're taking off a carbon, just like when we generated our acetyl-CoA, we're going to generate some carbon dioxide because that carbon molecule has to go somewhere, okay? So we're going to generate some CO2 at this step. And what we're also going to have happen, all right, is we're going to start sucking out some of those electrons out of this process. Because one of the key things of the Krebs cycle, or the citric acid cycle, is going to be to generate our NADH, okay? Because that NADH is going to be what gives us our electrons for our next step in cellular respiration. So it's going to give us the electrons for, oh, and you can't see that low, so I'll just write it over here, for our electron transport chain. Okay, so that's one of the really important things that happens with the Krebs cycle, is we're going to generate a whole lot of NADH. That NADH and those electrons are going to fuel oxidative phosphorylation so that we get the bulk of our ATP, okay? So we generate NADH here with the removal of that first carbon, okay? We generate carbon dioxide. That gives us our 5 molecule alpha ketoglutarate, all right? Then what we're going to do, all right, we're going to proceed in our citric acid cycle. We're going to lop off that other carbon. So that's going to give us another CO2 molecule, all right? So with that, 1, 2, this is our 2 carbon dioxides, okay? And what we generate from alpha-ketoglutarate is going to be succinate, all right? And I've put a little asterisk here next to the 4 carbons so that you can tell the difference between this 4 carbon molecule and this one, okay? So we've got a 4 carbon molecule, but what we have there in the succinate is we've got some other stuff that we're going to take out of it, okay? Before I get to that other stuff, I'm going to finish with the stuff that we generate from going from alpha-ketoglutarate to succinate, okay? So, and as part of that process, again, we are going to have NAD that's going to be converted to NADH, all right? And we're also going to have, in this step, we are going to have ADP plus a phosphate, and I'm doing this in pink just so you can see the difference here. That's going to generate our ATP molecule, okay? So what we've accounted for so far, we've got our 2 carbon dioxides, we've got our 1 ATP as part of our citric acid cycle, okay? But there's still more electrons that we can draw out of the molecules here in the citric acid cycle, okay? So what we're going to do then, going to succinate from succinate, we're going to go to malate, all right? And what we're going to have generated between succinate and malate, we've got another molecule here that I haven't talked about yet, all right? Don't let that throw you, all right? We've got FAD. FAD functions a lot like NAD. It carries electrons. It's just a different enzyme that will do that, okay? What we have happen here now is we get FAD converted to FADH2, okay? And that's going to also feed into our electron transport chain, all right? So the FADH and the NADH are going to be what bring electrons to our electron transport chain, okay? So you can kind of think of this for this level, all right? I'm sure there are people who would be horrified that I tell you you can do this. But this is just another way to carry electrons. It's another molecule, okay? A little bit different, that's okay. NAD versus FADH are going to produce different amounts of ATP out of them, and we'll talk about that when we get to the electron transport chain, all right? But in removing those electrons, we go from succinate to malate. Again, the star is added here so you know that this 4 carbon chain here is different from our 4 carbon oxyl acetate, okay? Because we still have a little more NADH to pull out of this, okay? Because right now we've just got 2 NADH, and I told you there are 3. So guess where we're getting our next NADH from, okay? When we go from malate back to oxyl acetate, all right, we are going to have another NAD come in and generate another NADH, okay? So in terms of what we've done here, we've accounted for our FADH here. Now we've got 1, 2, 3 NADH, and we've come full circle, all right? We now are back at oxyl acetate, which will remain in the citric acid cycle until the next molecule of acetyl-CoA comes in, okay? So what we've got here, 1 turn of our Krebs cycle is going to give us 2 carbon dioxide, 1 ATP, 3 NADH, 1 FADH. Because we have 2 acetyl-CoAs generated in glycolysis For each glucose molecule, we're going to make 2 turns, all right? So that next acetyl-CoA would come in. We lose the acetyl-CoA, where that can then be reused within the matrix of the mitochondria. That acetyl-CoA being lost, now we are going to join oxaloacetate to the 2 carbons that came in to generate our citric acid again, okay? Citric acid will be converted to alpha-ketoglutarate. We generate NADH and CO2. Alpha-ketoglutarate is generated into succinate when we lose that other carbon. Lose the carbon, we get carbon dioxide. We're generating more NADH. This is also where our ATP is generated in the Krebs cycle. Succinate to malate. Now what we're going to do is we're going to strip all of the electrons off of succinate. So we get electrons in the form of FADH. Succinate becomes malate. Malate, we lose more electrons or strip more electrons out, generate oxaloacetate. That's the citric acid cycle, okay? Those are the components I want you to know. I want you to be able to recognize this on a diagram. I'm not going to ask you to draw this from memory, but I may ask you where each product is made in the steps of the citric acid cycle, okay? And what I want you to take away overall from the citric acid cycle, really the point of this is not to generate a whole lot of ATP, because we don't get a whole lot of ATP. We don't get any more ATP out of the citric acid cycle than we did out of glycolysis. The point of that is to generate our electrons here, because the NADH and the FADH2, that's going to feed our next step. That's going to feed oxidative phosphorylation. If you have any questions, please let me know.